Hello, my name's Caden Pate, and we're going to talk a little bit about class switch for a minute. So, what is class switch? So basically, it's just the ability of a B cell to change the isotype of its immunoglobulins. So that's like from IgM to an IgG, or IgA, or IgE, whichever immunoglobulin. This type of post-transcriptional modification. So, how does it work? So, first you have to have the activation of a mature B cell, and you have to have some sort of VDJ recombination, and this results in the constant uh, portions of the heavy chain is changed, but the variable portion stays the same. So, that's just meaning that the antigen specificity stays the same, uh, but it's just able to act better. Like meaning, so you got a primary response, and in this primary response, you're going to have a lot of IgMs, and they're good at bonding, but they're not as good as IgGs. Meaning, an IgM is a pentamer, and to be able to bind up antigen, it has to have a higher volume of antigen present for those Fc regions to pucker and be able to have a response. Furthermore, that would mean that the IgG would be better. So it's through this class switch that these B cells are actually able to be able to bond stuff better. So continuing, a little backstory. So the naive mature B cell is going to express both an IgM and IgD. And this IgD kind of, we were studying about it and we just have it memorized that IgD is just something with B cell development. So it turns out that this IgD is again part of that B cell development, and it's on that naive mature B cell, but when it becomes activated, the IgD eventually falls off, and you're able to have that post-transcriptional modification to change the IgM to IgG. And we have to have AID for this to happen. So what is AID? AID is this activation-induced cytidine deaminase. And basically what this does is make uracil into, or cytosine for uracil. So you'll have these uracils in there where they're not necessarily meant to be. And that just is kind of causes these mismatches, and it makes it easier for the folding of that VDJ recombination to make a new isotype. Um, it just kind of tells it where to cut easier. It makes it better regions for the post-transcriptional um, modification. So that's basically what class switch is. It's the process of taking this B cell that is producing these IgMs and getting them to produce something better. So here's a little picture on it, and we can see that this T helper cell is going to go to the B cell, and the B cell is going to be asking permission to proliferate. So for it to be able to make all of these right here, you have to have that class switch, and they're going to be able to do different things in different ways better. And that's basically all what class switch is. Continuing, we're going to talk a little bit about tolerance to self-tolerance. So the central tolerance is basically the negative selection of both B and T cells. So these B and T cells will not need to be autoreactive, meaning like if they were going to attack yourself, that would be very bad. So it's through this negative selection in both that we're able to weed out the ones that are going to be autoreactive. Then you have something called peripheral tolerance, and this is just a little side note. It's just that this is able to have another kind of a checkpoint of saying you're not going to be autoreactive once you have exited these primary lymphoid organs on the way to secondary, and before it gets to even the periphery of where our T cells and B cells will be. And it's just another way for our body to be able to make sure we're not going to have any autoreactive cells. So just in the B cell, you're going to have negative selection in the bone marrow. And in this negative selection, uh, if it is autoreactive, you have basically three responses. Let me change that right there. That's anergenic. So the autoreactivity, if it's autoreactive, you're going to apoptosis. Or the B cell's going to say, okay, let's do some mix and matching and change this receptor so it doesn't recognize self. Well, it doesn't, not that it doesn't recognize self, but it recognizes self in the fact that I'm not going to kill other cells. And then the anergic state is just basically I'm not going to be able to do anything. I don't have uh, a license to kill per se. So continuing, I'm gonna keep on scrolling. Here is a good picture. So in the bone marrow, you have all these different B cells, and they're good. They are not binding self, but this little guy right here binds self. So once again, you have three different options. Kill yourself. You can change the way the receptor, or you can go in a state where you're just blah, I ain't not going to do a single thing. So let's go into T cell tolerance. So in this positive selection and negative selection, we're mainly worried about this negative selection, but the positive selection is important in the whole maturation of T cells. So the positive selection is going to be the ability of, the T cell being able to bind the MHC peptide complexes. And that's going to be in the thymic cortex. But it's going to also be for MHC class 1 and class 2. So, and if it goes through positive selection, it's able to, recognize the peptide MHC complexes. Fine and dandy. Let's go to negative selection. Are you autoreactive? If so, T cell, you're dead. You're done. So if it checks out all these things, it's either going to be a CD4 or CD8, and that's where it's going to go to the secondary lymphoid organs as mature naive T cells, meaning that they have not bound antigen yet. So that is basically all I have for class switch and self-tolerance.